Okay, everyone, I might kick off the seminar now, if I could. It's just a couple of minutes past 3:30. Welcome back to the CAR seminar series. This is the second one in the semester 2 lineup. Very excited to introduce Ray Tobler today. Before I do, I'd just like to acknowledge the Ngunnawal and Ngambri people on whose traditional unceded lands we meet today on this fine spring day in Canberra. Before I introduce Ray, just a couple of quick notes. The recent seminars for this series, including last week's one and today, will be available up on the ANU CAR YouTube page, probably this weekend or very early next weekend. You can get to it just by Googling, or in YouTube, sorry, putting in ANU CAR and it should come up. As I said, this is the second one, but typically the seminars are going every fortnight. The next one after this, after a short break, is on the 22nd of September with Peter Dowling talking about how epidemics nearly wiped out Australia's first people. And we'll send out more information about that closer to the time. And that again will be online. So if you do want to sign up to the newsletter, if you heard about this through other means, please do get in touch with myself or Matthew Leclerc to sign up, and it sends out information about various other seminar series and events that are going on. Or if you'd like to present a seminar in a later series, please do get in touch. We're very supportive of students presenting their research, so it's always a good venue for doing that. As always, of course, just please keep your microphones off. What I'll start doing is transferring people across to the panelist list. So at the end of the talk, we can ask questions and have a bit of dialogue and discussion afterwards. So it's with a great pleasure that I introduce today Ray Tobler. Ray is a population geneticist who's recently joined the team at ANU earlier this year in the School of Culture, History and Language as a ANU DECRA Fellow. So I'd like to very warmly welcome him to the team. He's also a key member of what is a relatively newly established Evolution of Cultural Diversity initiative within this school, which is a transdisciplinary team led by Nick Evans, of linguists and archaeologists and anthropologists, geneticists and philosophers who seek to combine the strength of the different disciplines to investigate questions around cultural diversity in New Guinea, the Pacific and Australia as well. So within this, Ray's research aims is to, as a population geneticist, is to reconstruct the largely unknown human genetic history of Australia, which he's published some very important papers on, New Guinea and Wallacea, which we'll be talking about today, using a combination of modern and genetic ancient human DNA genetic techniques. And of course, working closely with archaeologists and other disciplines to address some of these long established problems in the history of this broad region. So without further ado, Ray, I'll pass it across to you and take it away.
Thanks, Ben. Yeah, sorry everybody that you can't see me. I'm not sure that's such a bad thing, but if you do want to have a look at me, I'm sure you can Google an image and see what I look like. But basically, as Ben mentioned, I've been in ANU now for about a month, but I've come over to work with the ECDI, which is really exciting. I'm a geneticist by training, having done my PhD in Austria, in Vienna, where I was actually working with Drosophila, doing in a field called experimental evolution, which is a bit different from what I do now. And I moved to Australia in 2016 to take up a position at the Australian Centre for Ancient DNA, where I have been working on a project called the Aboriginal Heritage Project, which is ongoing. We had a paper out in 2017, which was published in Nature, which was fantastic, which was also using mitochondrial DNA, which is the basis of the molecular basis for the talk I'm going to give you today on Wallacea. And it just reminded, I think, a few people of the power of mitochondrial data now that everything's moving towards genomics, and I will talk a little bit about that. And certainly genomics is definitely a lot more powerful than mitochondria, and you'll see why during the talk. So I'm going to give a little primer on phylogeography and population genetics. But yeah, when you don't have any information about a region, as as is the case for Wallacea, for the majority of Wallacea, then mitochondria can still be extremely informative. It can often throw up a few surprises as well. So today I'll be talking about some of the work I've been doing with my colleagues in Indonesia and in Australia on the history of Wallacea, and also the relationships between Wallacea and New Guinea and Australia, the human history going way back to 50 or 60,000 years ago. Yeah, and some of the things that we've found so far. So we had this talk is based on a paper that was published, I think, about a month or two ago in a journal called Genes. And so if you're more interested in knowing a bit more about what I'm talking about today in a bit more depth, and some of the methods, perhaps, if you're interested in exploring them a bit more, then I recommend looking at the paper. So the paper is actually led by a PhD student of mine called Gludug Purnamo. He's actually from Indonesia. He's from Java. And he's the person who's really made this project possible. He's previously from the Eichmann Institute. He still collaborates with the Eichmann Institute. They're a genetics-based institute based in Jakarta, which have an ongoing project looking at the genetic history of Indonesia. They're really interested mostly in the medical stuff and medical questions, but of course, to really develop appropriate medicines in such a diverse population, you really need to know a bit about the population structure as well. So, okay, I'll get into the talk now. I'm going to go, as I mentioned, I'm going to go over a little bit of phylogeography at the start of this talk, just to give people a bit of an idea about how we geneticists go about actually extracting information out of genes and genomes that tell us something about the past. So hopefully it won't take too long. And if at any point, I don't mind if people have a question, if they want something clarified during this part of the talk or any part of the talk, please just let me know. I probably won't see if you put the hand up or whatever, and the the little icon. I think it's just easy if you just ask. So once we've gone through a little bit of background, I'll go into the key results from the current project in Wallacea. Doing a little bit of background on that as well, and then where things are going in the future. Okay, so like I said, in the last 20 years, we've had a deluge of genomic data being produced since the production of the first human genome around about 20 years ago at the turn of the millennium. Since then, the technology has advanced really, really rapidly, and it's got to the point now where you're seeing thousands, tens of thousands, if not hundreds of thousands of genomes being produced in certain parts of the world. But before that, since about, I think, the 70s or 80s, most of, when people were interested in looking at the genetic history of a particular population, they would use either a single marker, or such as the mitochondria, or a gene or a genetic variant in the genome. And the mitochondria was used most frequently because it's actually, unlike the full nuclear genome, it sits outside the nucleus. It's manufactured in many copies in every single cell, and it's quite short. So whereas the genome is around about 3 billion base pairs in humans, mitochondria is about 16,000 base pairs. So that's the nucleotide sequence that makes up the mitochondrial genome. So sequencing the mitochondrial genome has been possible for a while. Used to still be quite expensive. Now it's just rudimentary. And it provides genetic information, obviously, for each individual. Every single individual has a mitochondrial copy of a mitochondrial genome that can be used to learn about history. And what that image that you're looking at there is from, I think it's, I've got it down the bottom there, from a paper in 2015. Where they basically compile all of the mitochondrial information from the major mitochondrial lineages from around the world, and they plot it on a map of the Earth, of the occupied continents, showing how that diversity, that mitochondrial diversity, is structured with respect to geography. And so what you're looking at there is effectively a tree, which is relating all those mitochondrial sequences. And the more closely those leaves are together, or the tips of the tree, which are called the leaves, the more closely related those populations are. And so you can see for instance, the really deep parts of the tree, although it's not so obvious in this plot because the tree actually curls around to fit in with the picture, but the deepest branches all go back to Africa, and those are what are known as the L lineages. And then a subgroup of those L's are all the other branches that radiate around the rest of the world, and those are split up into major haplogroups. So each of them just gets a name. They don't really have any sort of quantitative basis. They just sort of a legacy of how these things were discovered sequentially in the past. But each one is sort of marking out a set of mutations that are distinctive to those mitochondrial lineages. But the key is that the M, N, Rs, H, Vs, all the other non-L haplogroups or lineages extend from one branch of the Ls. And this was some of the first evidence that non-African human populations are a radiation of African populations. So you've still got all that ancestral diversity in Africa. And then a group of individuals got out, a small population, we don't really know the specific details, at some point, and that radiation or that early migration into Eurasia is what seems from genetics at the moment to be the basis of all non-African human populations globally, which is quite remarkable. One thing to note is that there probably were earlier movements of anatomically modern humans into Eurasia and beyond. I think the evidence is probably pretty good for that, but it doesn't look like they successfully left offspring that have lived until the present, and that's a really interesting result in and of itself. So, and all of this was possible just by working with mitochondrial DNA. So you could get the major sort of picture, the global picture of what genetics had to say about human diversity and relationships from mitochondrial DNA. Of course, it's since been reinforced with the Y chromosome, which is the inherited paternally through the male line, and also the full nuclear genome as well. And with the nuclear genome, you can reveal much, much more. But the broad picture is the same. Oh, wait, why? There we go. Okay, so as I mentioned, there's the mitochondrial DNA is, well, I may not have mentioned, is a single non-recombining unit, meaning that it is one sequence that is inherited as a whole from, in this case, your mother. And what I've got here, what I'm showing here in this picture is basically a pedigree. So where you can imagine yourself as the ego there at the bottom of the image. And as you go up one generation, you've got your parents. And of course, you've inherited your DNA from your parents. Half has come from your mum, half has come from your dad. But the mitochondria comes only from your mother. Right, so that's a maternally inherited DNA. It doesn't recombine, meaning it doesn't get reassembled in your mother's gametes before it's passed down, whereas the nuclear DNA does, because it has to half each generation. So what actually happens is this process called meiosis, where chromosomal DNA, nuclear DNA, it actually you've got two copies in every cell. It reassembles some parts of one copy will shift across to the other part and vice versa. And this basically shuffles the nuclear genome, but this does not happen for the mitochondrial DNA. Right? The mitochondrial DNA is a single unit that does not recombine. So I mentioned the Y chromosome is a paternally inherited, sort of the equivalent of the mitochondrial DNA. That comes through the father, but only males have the Y chromosome because it is it contains the sex determining locus of the genome. So you get a Y chromosome. Well, most of the time if you get a Y chromosome, you're going to be male. But if the sex determining locus actually jumps off the Y chromosome, which does happen occasionally, then you can have a Y chromosome and actually not be male. So, but the key is it's actually inherited paternally. Mitochondria is inherited maternally, and the nuclear genome is inherited through everybody else. So you can see there that mitochondria only captures that one lineage. It goes from mum to mum's mum and so on through the past. It's entirely capturing female history. It never is ever situated in a male where it is inherited. All right, so it doesn't capture any information about what males have been doing in the past. So that's an important caveat about mitochondria. And that's why when you talk about mitochondrial history, you talk about mitochondrial Eve. You may have heard that term. That's because when you go back to the common ancestor of all current copies of mitochondria segregating in the, in every human population in the world, they come from one theoretical ancestral female way back in the past. And yeah, people have tried to date this. The same, you can do the equivalent for the Y, and that goes up that paternal line. I hope you can see those green arrows. They're not really standing out that well. And the cool thing about the genome is that captures the ancestry. It's not a single lineage. It captures ancestry from all of your ancestors, but in finer and finer proportions as you go further and further back in time. So where you get half of your DNA from your mum and your dad, guaranteed. From your grandparents, you get about a quarter each. Great grandparents, about an eighth each. And as you go up, obviously it doubles each generation. As you go up through successive generations, once you get to the eighth generation back, great, whatever grandparent, you're only getting about 256th of each of that generation is in your genome. And actually you can have, and this is one of the cool things about genetics, because of that shuffling process in the nuclear DNA, you can have biological ancestors where you have no genetic information from, because there is a chance that it just gets lost in that shuffling process because not all of it is passed on. So your genome is a mosaic of all that ancestry, but it doesn't capture everything, and it's slightly unbalanced. On average, it will capture about those proportions in each generation that you see on the screen, but sometimes it gets lost. So that's another sort of interesting thing about the nuclear genome. The crucial thing, what makes it really powerful, is that recombination process shuffles all those different histories, and each one is effectively capturing an independent component of that history. Well, they're sort of correlated along the genome, but if you move far enough along the genome, you're going to get a completely different ancestry. So whereas a mitochondria gives you one history, you can build one genealogical tree or phylogenetic tree. In the genome, you can build thousands, tens of thousands, hundreds of thousands. And there's all these tools out there now that are being developed where we're actually trying to infer the whole recombining history of a genome, of populations of genomes, to get thousands and thousands and thousands of these trees. And that you can get, for instance, just a couple of or one individual, because you've got 2 copies of each chromosome. You can get all sorts of history much more history that you could ever recover from a single, or from even, quite a few mitochondrial genomes. All right, so what do we do when we have, for instance, mitochondrial data from a population? And this part of the talk, I'm just going to really, for about 10 minutes or so, go through, try to build an intuitive understanding of the basis of inference in phylogeography. So first of all, how do we generate a history, which we call a phylogeny? It's technically, if you're talking about a populations, it's a genealogy, but the term phylogeny really comes from earlier in genetics, when we're talking about species relationships more than population relationships, but these things are really just a matter of scale. There are some things that, of course, differ between populations and species. Populations are able to sexually reproduce, for instance, and that doesn't happen across species per se, but the general themes are the same. The idea is that you're reconstructing a tree that represents that genealogical phylogenetic history, how all these individuals are related. And what you're trying to do is infer that process from genetics. So what I'm showing here is, you can imagine that for each of those people depicted at the bottom of the screen, each of them carries a mitochondrial genome. Their genomes have been inherited from their ancestors, so most directly their parents, but in this case their mother, because it's mitochondrial DNA. When we take any sort of combination of people, at some point the copies of mitochondria that they carry have a common ancestor, that each copy had to be replicated at some point. So we can actually trace back from any pair of people a common ancestor where that copy was actually first replicated, right? And you can do that for all people, and you can build what you have here, which is an ancestry. So in this case, what we have is a known ancestry, right? So this is actually hidden from us. This is what we're trying to infer, but this is what we're showing is, this is what we know, right? This is the true genealogy of these particular individuals. So how will we go about trying to recapture that genealogy? Well, one of the nice things about DNA that allows us to do this, and why we actually have biological diversity at all, is because, of course, it mutates. That replication process is imperfect, and it introduces new mutations, and those new mutations occur in certain parts of the genealogical history of a sample. So as by way of sort of showing how that works, if you can imagine that ancestor, that most recent common ancestor, so that's the individual that carried the mitochondrial copy, that is basically the ancestor of all the copies and the people at the bottom of the page there, the screen. And what I'm showing is like an idealized representation of a DNA sequence. It's got 5 different positions of the ancestor. They're all colored white. And then as we move down the tree, we're seeing mutations occurring at certain points in that history. The key to all of this is that at the bottom, if you trace any of those individuals backwards through time along that tree, and you just look at where those mutations are occurring, that creates the haplotype, what we call a haplotype that individual carries. And then what we can do by looking at the specific mutations that people have in their mitochondrial DNA, we can then, based on the notion that it's inherited and it must share some common ancestry, we can look at the differences and actually group people together. So we could try to rebuild this ancestry, or at least the topology, the relationships between the individuals, just by looking at those mutations. Here we've only got 5, but in a mitochondrial genome where you've got 17,000 different positions, obviously there's going to be tens to hundreds of informative sites that you can use to build these trees. The other thing that we can do is that these mutations actually accumulate at a certain rate. That rate can then, by counting the mutations, we can actually extrapolate the timing of these events, because between any 2 pairs, you can count the number of differences and you can work out how long it's been since that came from a single copy, because all those differences must have accumulated since that copy first replicated. So we could get a rough idea. It's actually a pretty good idea of when these haplotypes actually coalesce, what we call coalescent process, when they trace back to a single common ancestor. And then we can learn about, for instance, when certain groups of individuals last had common ancestors. So this could be 2 populations, 2 language speaking groups, anything you can imagine that would be of interest to understand the relationships that extends beyond single individuals. So the next key step, what takes phylogenetics to phylogeography in sort of the coarsest sense, is just looking at these patterns in the context of geography. And so what I've done here is just taken the genealogy that we saw before, and I've tipped it on its side. And now you're looking at, instead of thinking about individuals at the bottom, maybe the families or groups or populations of individuals that are related according to these, or we've learned the relationships according to their mitochondrial DNA. And so we build that ancestry, and then we say, okay, here's the tree. Now, where are these groups of individuals in a geographical context? And does that tell us anything potentially about how these groups are related to one another, and something potentially about how they managed to get where they are today? So information on demographic and historical population processes. So for instance, we might go out and type a whole bunch of individuals around Australia, just using Australia as an example, and find that for those people who have this particular type of mitochondrial DNA, we only find them in a certain region of Australia. Likewise with these other 2 groups, they happen to occupy fairly discrete other regions in other parts of Australia. And then similarly for this group here. And what we're basically getting a picture of here is that we've got very distinct structuring genetically that is also correlated in some way with geography. And in this case, we might say, well, it looks like we've got what we're calling the A families, A1, A2, and A3, seem to be confined to the East Coast. The other families to the west and moving around to the south. And maybe this is telling us something that earlier on in the common ancestry of all these groups, there was a movement into Australia. One branch, the green branch, the A's, went east. The other branch, the blue branch, went to the west and south. So this would give you a sort of a coarse first approximation of population processes that might have occurred historically to generate the patterns that you're seeing in modern populations. So that's all good and well, but of course, this assumes, and this is a really important assumption, that the location of people today tells you something about where they were in the past, right? Because those populations today could have been anywhere or their ancestors could have been anywhere in the past. They could have moved there 2 weeks ago, right? And we would not necessarily know that just by the DNA does not tell us that. All the DNA tells us are the relationships with other units of DNA. And that's why we need to talk to archaeologists, anthropologists, linguists, and everybody else to actually get a much firmer understanding of the relationship between modern populations and historical populations. So I just want to show you here that you might have, for instance, what is very common today, populations starting to move around, expand a bit more. And what that means is you don't get these nice discrete relationships with geography. It's still there, but now it's much, much coarser. So you got to work much harder to actually reconstruct the past. And the key for this, so genomes, moving from single copies to genomes is one way, right? It provides a lot more information, but it still doesn't help if people have moved around to such an extent that there is no longer any information from the present that relates to population locations in the past. The key really is ancient DNA, because ancient DNA puts genetics on the map in a particular location historically, so that you can, especially if you're getting a lot of the same haplogroups in a particular place historically, you're getting pretty sure that this is where people were at that point in time. So it actually allows you to build much stronger understanding of population of processes that have occurred historically. Yeah, so the other thing is sometimes so that's an issue. The previous slide was talking about movement of people, which can confound inference, but the other big issue is actually the inference procedure itself. So the trees that we generate, so these phylogenies, may also not actually represent the population history. And just to showcase this, we could have built this tree. So if you go back before, remember we had this tree before, but we could have equally built this tree based on the mutational information that we have here. And that would give us a slightly, different scenario in how we might interpret historical events leading to where these populations are today. Okay, so does anybody have any questions at that point? Because that's basically phylogeography in a nutshell. But I'm happy to actually, if you've got anything you want me to clarify, anything at all, please just stop me and ask, because if no one has any questions, I'll just keep going. But if there is something now, I'm happy to attend to it now, because other people are probably thinking the same thing, wondering the same thing.
Ray, there's just a quick question here from Liz.
Cool.
Hi, how accurate is it these days of estimating the rate? Because I understand that decades ago the rate was set by archaeologists, and then archaeologists used the rate to set archaeological time. I assume it's a lot more accurate now.
Yeah, no, they're definitely improving. So that's not an area that I work with directly, but we've got some good people at ANU actually who do that. Yeah, there's all sorts of caveats with inference and mutation rates. So you get differences across the mitochondrial genome. So you got regions of the mitochondrial genome that are functional, that produce proteins. They are less prone to accumulate mutations. They're under a process called purifying selection, where you actually weed out most mutations because they don't improve the mitochondria in any way. They make it worse. Other parts of the mitochondria are more neutral. You've got one region called the control region, which is highly variable. And that was the region of the genome, mitochondrial genome, that was used historically. And that's the fact that it's so variable means the mutation rate is higher. It's good for looking at more recent history. You don't want to be using a part of the genome, for instance, that hardly mutates, where you've got the strong purifying selection. If you're looking at, history over the last 20, 30,000 years for humans, for instance, but that's really good for looking at deeper time. Because the other thing is, if you've got really high mutation rates, then you get this thing called saturation, where every nucleotide is mutated so many times it no longer recovers anything about the history anymore. So you've got to balance that out. So humans are in a bit of a sweet spot in terms of our history, where mitochondria is actually quite useful. We've got a much better idea now about the mutation rates from, pedigree studies, and there's many, many different ways you can infer it. There's still outstanding questions for sure, but it's improved to the point where inference over most of the time periods that we're interested in is pretty reliable. The other side of it is that mutation rate can change through time or along a specific lineage. That, I think, is not as well resolved. That might require ancient DNA to work out a bit more. For instance, the mutation rate that we've inferred might be really good for the last, say, 30, 40,000 years, and then get a bit wonky before that. But then you've got to start thinking of scenarios about why that might be. And it seems to work pretty well. It seems to line up with the archaeology fairly well, but although archaeologists might tell me otherwise. So yeah, and the way it used to work in the past, of course, was it was calibrated as well. So you would have certain parts of the tree where you could fix the depth of that part of the tree using archaeological dates as sort of an upper or a lower bound. So I'm sure they can still be used interchangeably. There are definitely caveats that you need to think about, but I think the thing with mitochondrial research, like a lot of the dates that have been coming out, say the last 10 years, when the inference has got a bit better about the mutation rates and tools have got a bit more sophisticated to handle variation in mutation rates, both within the chromosome and through time, they line up quite nicely with inferences from the Y chromosome, which is also a single non-recombining unit, or part of it is. So they work pretty well. They line up quite nicely. And now with all the nuclear data, it's also sort of fits in pretty reasonably well with the nuclear data as well, which you can use a whole bunch of other techniques for timing. You don't have to rely on mutations. You can, for instance, use recombination as a way of inferring the past.
Great, so thanks. That's really good. Thanks very much.
Yeah, no worries. I'm just, I worry if I might run out of time, but I really want to, like, one of the main reasons I came here was I really want to start working. I know nothing about human history other than, the few books that I read as an undergraduate when I still had time to read these things. And I've been working on these projects for a few years, and to really understand human history, it's not just all about putting genetic points on a map. I really want to work more closely with archaeologists and people who work on human history to help understand and just even frame hypotheses. So it's just, and to do that, I think we need to start understanding some of the, building an intuitive understanding of these methods. Obviously the nuanced stuff, it takes a long time to build up, and that's why we specialize in our fields, but most of the stuff I think is quite understandable. And there's just so much opportunity to do it now in ANU. So yeah, no problems. I'm happy to answer any questions, and also after the talk. But yeah, so getting on to the current focus of the talk. So I've been working for like the past 4 years now on a project which was initially kicked off by wanting to understand more about the peopling of Sahul, in particular, not so much the timing, more the routes that people had followed. And again, this is where phylogeography and latterly population genetic techniques could be really powerful. Because you can imagine, well, as you can see here, you've got the map of the region with also the the continental land masses over different time periods. So going back to 60, 70,000 years ago, when obviously the water levels were much lower, sea levels were much lower. And one of the really cool things about the fact that Sunda, or mainland, what was then mainland Eurasia, and Sahul are separated by a bunch of islands, is that you get all this genetic diversity sort of accumulating on each of these islands, right? And you could imagine a scenario where people, and of course it's not actually like this, but people obviously had to move from Sunda across Wallacea into Sahul. And in an idealized scenario, what would have happened is those people just stayed on those islands, and their genetics evolved independently on each of those islands. And then what we could do is we could go out and we could survey as many of these populations as possible across each of the islands. And you could look at the relationships from people living now in indigenous populations in Papua and Australia with the indigenous populations in Wallacea. And you could basically work out, by looking at the genetic relationships and then framing them back onto the geography, the probable paths followed by those groups. Now, that was the idea when we started the project. We thought, okay, we'll give this a go. We'll start with mitochondrial data. So through collaboration with a team at the Eichmann they've generated thousands and thousands of samples. We had a PhD student who came over and started a PhD a few years ago. And to start it off, we started, before moving into the genomic data, which we also have, we thought we'd just start working with the mitochondria just to see what we could see, if there was anything that would pop out by looking at the mitochondrial DNA. But that was the framing question, was, can we say anything about these different migration routes into Sahul? And of course, you guys are no doubt very aware of the different debates about the possible routes into Sahul, so I'm not going to go into detail about that here. But just to say that the two more prominent ones were one moving through the north through Sulawesi and the Moluccas, and another moving through the Lesser Sundas in the south. But of course, any combination of these routes are possible, and it might not have been a single landfall. It could have been multiple movements through time. And in fact, there was a paper recently, I don't know if anybody's seen this, in MBE, which is actually a genetics journal, so it wasn't, might not be that well known outside of geneticists, but it was a study of maybe 60 genomes in New Guinea. They combine it with other genomes that are now available, and they try to work out a bit more about this migration process. But of course, the key, and they say a few interesting things, I'm not sure I agree with everything, but the key is that they're still missing a lot of data from Wallacea, and this is what we have for this project. Oh yeah, so the historical movement of people into Sahul, that's what I've mentioned previously. And as I going right back to the start of the talk, it looks like this is one branch, which is really remarkable, of a worldwide dispersal, taking some place between 60 to 50,000 years ago, where people have moved through the Southeast Asia into Sahul. And some of these groups have moved up into the Philippines, and genomic data has basically established this model that this is basically accurate for these original movements. And another branch has come into Sahul. And then more genomic research in the last, I would say, 5 years has shown that, of Indonesian populations, has shown that they're a complex assemblage of different ancestries, some of which is this original movement of people, some of which comes from what they were calling in the original paper from about 4 or 5 years ago, an Austroasiatic contribution. And if there are any archaeologists here, you may know much better than me what groups it is thought that these groups are, but they didn't, apparently, according to the genetics at least, there's no evidence that any of this genetic contribution got beyond Sunda. So there's no evidence that this Austroasiatic contribution is in the ancestry of modern individuals sampled in Wallacea. Of course, we know nothing about northern Wallacea up until very recently, this paper being one of the first looks. This is based mostly on the Lesser Sundas. So this is these Austroasiatic people. So this would have been around this is certainly Neolithic period. And then more recently, well-known Austronesian movement, which is still debated exactly where these people came from. Again, it could be numerous places, but they have share the closest ancestry in terms of the genetic components and populations now worldwide. The closest living genetic groups are Taiwanese, indigenous Taiwanese people. And this movement resulted in all sorts of mixing and movement of people around the Pacific, Indian Ocean, and contributed heavily to Indonesian populations. There was some contribution also in New Guinea. Open question, did it happen in Australia? So far, with the very limited evidence we have, no. Torres Strait Islanders, yes, it seems so, but again, we don't have a lot of information from Torres Strait Islanders, only I think mostly mitochondrial information. But all of these movements, I should go a bit faster. And there's also some possibility of back movement from New Guinea that has been suggested based on some of the patterns genomically, but not substantiated. And that's important for what I'm going to discuss in this talk. But all these movements sort of have resulted in these clines of what we call clines of ancestry, where as you go from west to east, from island Southeast Asia, Indonesia, across into New Guinea, you basically get an Asian ancestry decreasing from the east. So what's that, Sumatra, running, no, from the west, sorry, from west to east. So from Sumatra across into New Guinea. And there's a counter gradient of what is sometimes called Papuan ancestry, what sometimes is called Negrito ancestry, Negritos being pockets of small hunter-gatherer populations dispersed throughout island Southeast Asia that are thought to be modern relics, basically, of those original movements. But no one really knows. There's different hypotheses. No one really knows for sure. But the main sort of takeaway is that you do seem to have more Asian ancestry as you move west, more what we call probably original ancestry from that first movement as we move towards the east. Yeah, and the big question is that we started out with is this really going to, if we look at these Wallacean populations, if we get more information from these island groups, can it tell us anything about the peopling of the region? Can it help resolve some of these questions that we have? And we had a sample of, I think it was 360 individuals' mitochondrial genomes. And from those, we extracted, I think it was something like 180 of them, about half, 120. Oh no, sorry. So there were about 300 new samples, 120 of them from West Papua. Of course, that's part of modern Indonesia. The rest of them are coming from the Wallacean islands that you're seeing there. Most of them are from northern Wallacea. We have one population in, I think it's Roti, in the Lesser Sundas. And from those 300 odd samples, we concentrated on only the mitochondrial groups that were not part of those Asiatic movements, because what we really wanted to see was the history that was related to the deeper ancestry of the region. And if you're pulling in these haplotypes from whether they be Austronesian, Austroasiatic, what have you, you're actually looking at the ancestries in those regions. All right, so we ended up removing about half, I think, or maybe a little bit more of those 300 mitochondrial genomes, and we just worked with the ones that were haplotypes that were closely related to Papuans and Australians and are not seen anywhere else in the world. We combined them with another 300 odd genomes from New Guinea and Australia, and then we built a phylogenetic tree using this software called Beast, which doesn't really matter too much, but the nice thing about Beast is it's a Bayesian method, so you actually get a posterior of trees. So you get thousands and thousands of probable trees, which you can then use to build consensus trees and make inferences. So that's what you're seeing here. So I always find these things pretty horrible to look at, and I've been looking at them for like 10, 15 years now. But the key here again is you're looking at relationships. Each of those at the ends of the tree, which is to the right of the screen, you've got all the different nomenclature for the different mitochondrial haplogroups. And those, again, they are somewhat arbitrary. But at that side of the tree, you've got the individual samples, the individual mitochondria, and then you're just going back and you're linking them to the most recent common ancestor. So that the closest so where two samples meet, at about that time, that DNA had replicated to become two forms, and that's where those two individuals trace an ancestor. And the further back you go in time, the longer it is that obviously that these two units of DNA have been traveling, right, independently. And what you're looking at here is we've colored each of the samples according to where they were collected, and that's either Wallacea, New Guinea, or Australia. And the cool thing is that the Australian DNA, which is something that we expected to see, and it turns up again here, forms very distinct clades. They are very deep clades, so they branch off from both Wallacean and Papuan mitochondrial groups right at the base of the tree, so to go to the left of the tree. And that's effectively telling you that those separations between what are now modern Aboriginal Australians and Torres Strait Islanders and Papuans and Wallaceans happened a long time ago. There are some other parts of the tree where you might find an Australian haplotype nested amongst, say, a Papuan haplotype, but they tend to be less frequent. So effectively, it's saying that those groups separated fairly early on, and there has not been a lot of movement or gene flow between those populations henceforth. There has certainly been a little bit, but not a lot. When you look at the Wallacean and Papuan parts of the tree, what you tend to see is that the Wallacean groups tend to be nested in amongst Papuan diversity, and there are not many parts of the tree where you get a really deep rooting Wallacean individual. You still see it occasionally for Papuans, they exist, those deep branches, but you don't see it much for Wallacean individuals. And that is sort of pointing to some relationship, some closer relationship between Wallacean individuals and Papuan individuals. Either they separated quite recently, or there's a deeper separation, and there's been movement of one or the other. So the more likely movement is the fact that most of the Wallacean groups are nested within Papuan groups, moving that there's probably a movement from New Guinea into Wallacea. And so we weren't expecting to see it, the extent of it. We thought we might recover quite a few deeper Wallacean groups. That was the hope, because only these really deep groups are going to tell us something about, like I mentioned before, if people had got to these islands, stayed there, hadn't mixed too much, then we would be able to recover some sort of phylogeographic signal about the movements through these regions. Turns out we weren't able to do that. And so the next question became then, well, what's going on here? What's happening? That was highlighted in yellow rather badly, I think, because I don't even know if it's sitting on there right, is this P star lineage. That is one of the deeply rooting Wallacean lineages. And I'm showing half the tree here. So maybe what I should also add, when you come out of non-African populations, all mitochondrial haplogroups are broken into two major branches. One is the N branch, and that's what I've visualized here. And the other is the M branch. So right at the top of that tree, we've collapsed all of the M's. That's probably more than half of the sample data were M's into this one group. And that what you're seeing there is the deep relationship. So when we go right to the base of that tree, that's the split between the M's and the N's. So the M's and the N's are found all around the world. They've migrated back into Africa. They arose in Eurasia. So they're part of those really early splits in Eurasia. And they also exist in Papuan and Australian groups, but that 70,000 year date, probably not that reliable. It's maybe, I think that's been better dated somewhere between 50 and 60,000 years. That's the really deep stuff. So that stuff was amongst, so that you can think about that being genetic diversity that was present in those ancestral populations as they came into this region. And anything actually anything between, say, 60 and 50,000 years, if you're seeing splits at that point, that may have happened somewhere in Sunda. It could have been in Australia. It's not too clear. We don't know for sure unless we go out and look in the Philippines and other places as well. There could be populations from there that root in this tree, which would tell you it was probably a split earlier on, rather than latterly in Sahul. So this is, I apologize for this sort of very busy image, but what we did next is we tried to understand a bit more about the population processes more recently in time by looking at what we call geographically exclusive clades. So looking inside that tree and just finding groups of individuals that share a common ancestor, but each one of them comes from the same place, right? So they're either from Wallacea, Australia, or New Guinea, with the idea being, say you had a group of 20 individuals and they all share a common ancestor, they're all from the same place, right? So that would tell you that this is probably a radiation of individuals in a particular part of the world. So we can trace that ancestry back to their most recent common ancestor and get an idea of when that radiation occurred. And so, for instance, you could extrapolate that to all of Australia and look at all of the radiations of these lineages in Australia and try to get an idea of when Australia was first inhabited. And that's what we did in our paper from a few years ago. And that's effectively what we're doing here. And on the, so you've got 3 panels, one for Australia, one from New Guinea, and one from Wallacea. What you're seeing is on the y-axis is sort of the distribution of times across all 10,000. So you've got all these 10,000 posterior trees from the beast inference, right? Each of those trees is one realization of that genealogical process. So there's some variation across those trees. And what we did is we took trees or these geographically exclusive clades where that clade was present in at least 50% of the 10,000 trees. And then we said, well, what's the date of the most recent common ancestor? And that's giving us some measure of what I was saying before, like when a radiation is occurring, or perhaps where it marks when a migration first comes into a particular place, and so forth. It's capturing that information. And what you can see is that for Australia, you had something like 12 of these distinct clades. So each of these geographically exclusive clades will be a unique set of individuals that always, or at least 50% of these trees appear as a clade. And then you're just looking at their distribution through time, right? And so that's effectively what you see for Australia. Papua is a bit shallower as you come to the central panel. Wallacea, shallower still. And so that's already telling us something, but the next key component is to aggregate all of that information and say, well, what does that tell us? What is the sort of the average information that this is telling us about historically? So we then took all those distributions for the individual clades, put them together, and then generated an overall distribution for each of those 3 regions. And what we're looking at here is the distribution of the most recent common ancestor for each of these geographically exclusive clades and what that looks in aggregate. And the really neat thing here is that for each of these groups, you're seeing these 2 fairly clear peaks, not so clear for Australia, and that's interesting in itself, but very clear for New Guinea and Wallacea. So in Aussie, the big difference is you've got this big deep tail going out to the right. So there's a lot more of those geographically exclusive clades where you have a very deep common ancestor. But you still have this peak at about 3,000 years ago and another peak about 16,000 years ago. The peak at 3,000 years ago comes from Torres Strait Islanders, right? There are 2 clades, where each with 2 individuals from the Torres Strait, and their next ancestor out is always Papuan. So this is something going on in the Torres Strait, seemingly. Then when you look at the same points in time in Wallacea and Papua, you get much more distinct clades. So that many more overall of the common ancestors for these geographically informative clades are coming from those 2 of that particular period, with another distinct point being at around about between 14 and 16,000 years. So this is where you've got to now say, okay, this is the aggregate sort of information from the genetics. What could this possibly mean in relation to historical events, geographic, climatic events historically? And so the two things that stood out were the shift in climate during the last glacial maximum, around about that period, 15 to 16,000 years ago. So the end of the last glacial maximum, no doubt lots of things were changing in these regions. In Australia, New Guinea, certainly the climatic records there are not too bad, and you do get a sense of how much things have changed here in those regions. Wallacea, not so clear. I'm not so familiar with the climatological work done in those areas, but it was interesting that you also saw the peak there. So what it's basically suggesting is that you've got a lot of radiations going on in these time periods, right? And the other one, of course, being 3,000 years ago, relating to the Austronesian movements into these regions. And so, okay, those are the sort of the proximal events going on at the time. So what might be happening? It became a little bit clearer from a paper in 2017, which was a genetics paper of New Guinea, where they showed that in New Guinea at the time, there was a massive demographic shift right across New Guinea. Well, at least in the trans New Guinea speaking populations. And it looks like the interesting thing is it looks like most of the population structure in New Guinea was generated around about that time. So although New Guinea had been settled for a long, long time, something interesting was going on 20-odd thousand years ago, between 10 and 20,000 years ago, where most individuals today in New Guinea would find wherever you looked in these populations in New Guinea, a recent common ancestor about 20,000 years ago, kind of suggesting there was a source population that radiated out to generate most of these trans New Guinea speaking populations that we see today. We've got West Papuan populations in there. I'm not sure if all of them are trans New Guinea speakers, they may be. But it seems to be that this was also happening on the West Papuan side as well. So there's something going on across the country. And there may have even been movements from New Guinea into Wallacea. That would explain why all the Wallacean mitochondrial genomes are sort of coalescing around that time, because Papuan women have effectively come into Wallacea, and then they have radiated in situ. Right, they've had children, their children have had children. So their common ancestry goes back to that point. That's where they find their most recent common ancestor. And then you get another wave going on 3,000 years ago, which looks like at the time of Austronesian arrival, there's another movement, and it's pretty much looking like it's moving in the same sort of way. We're seeing also an impact in the Torres Strait, which is interesting. So perhaps that means either Austronesians were mixing with these local groups, moving those mitochondrial haplotypes through the mixing and then moving, or the local populations perhaps used the technologies introduced by Austronesians, or something else was happening during that period that also motivated the movement of these genetic lineages. Okay, so that's effectively what we got from the genetics. I was lucky enough to be able to talk a bit with Sue O'Connor and Shimona Keeley, who I'm sure you're all familiar with. So we had all the genetic results sort of pulled together, and we thought this is pretty amazing. And then we got in touch with Sue and Shimona, and they had been working obviously extensively in the Lesser Sundas region, and had found a lot of, in their archaeological work, some of the records really point to either movement of or transfer in technologies around this point, so obviously dated in various sites across the Lesser Sundas, or an intensification of some of these sites, perhaps that hadn't been used previously, or there wasn't very much evidence for maybe 20,000 years in particular sites, and then a sudden intensification. Both at around about 15, 16,000 years ago. And maybe if they're here, I don't know if they are, they can speak to this as well, but also around the Austronesian movements as well. There's other lines of evidence as well. So there is this really nice connection now between what we're seeing in the genetics and what we're seeing archaeologically that's pointing in a particular direction. And that seems to be saying that whatever was happening in New Guinea during these time periods resulted in the movement of Papuan people, women, into Wallacea. It's affecting the top of Australia as well. We see some evidence in the Bismarck archipelago as well. So it looks like it's extending out of New Guinea, it's extending into the surrounding regions. Interestingly enough, there's a paper, genetics paper from the Philippines, where they show that, I think it was in Luzon or one of the southern islands in the Philippines, they also see Papuan ancestry cropping up. And they say it's somewhere in the last 15,000 years, interestingly enough. They don't try to date it. So it could have been even more extensive, the movements at that time. And so there's, of course, this leads to many interesting questions about what was happening at that period, what was driving it, and whether what actually happened in Wallacea itself. Were these islands no longer occupied? Was this effectively a recolonization? Was it a mixing event? Was it a replacement event? We don't know. Obviously, from the genetics, we don't know at this stage, especially just from the mitochondria, but these are all questions that can be posed a lot more powerfully with the full nuclear data. And that's where I'll end, just saying that that analysis is coming up. What you're seeing here in this image is one of the most remarkable things over the last 20 years. I mean, genetic data has just absolutely revolutionized population genetics, which was largely theoretical. I mean, you could work with mitochondria, you could work with single copy genes, but you have very limited power to really resolve the questions that most people were interested in. And that has completely changed in the last 20 years. So what you're seeing there is the change in price for a genome, a single genome. It went from, I don't know, what is that, 10 million? It was more than that. It was like 100 million or $1 billion to produce the first genome, down to the present period where we're talking about less than $1,000 for a genome. I think it's come down to around about 100 recently, and that's just going to keep going down. So there's just this huge amount of genomic data out there now, and now it's been catching up with methods. Unfortunately, there's a lot of really bright people working in the theoretical aspects and developing new methods. And I mentioned previously that we're looking at trying to get every recombining unit of a genome and a population of genomes to extract thousands, tens of thousands of trees to get all of the information that you can possibly extract out of genomic data to look at these histories. And that will just be a complete game changer for a number of reasons, especially getting really high resolution information for demographic processes, including up to recent times. And I'll just give a plug to my colleague, João Teixeira, who's also come across to ANU. I've been working with João for the last 4 or 5 years. He's interested primarily in the history of hominid interactions with humans as we moved into Sahul, as populations in this part of the world have contributions from what we call Denisovans, but we don't really know what they are. And unique contributions from these populations probably occurring somewhere in island Southeast Asia. And what João wants to do as part of his DECRA project, which he got funded as of this year, is use genomes to really reconstruct in high resolution, first of all, the different sources of this hominin ancestry. So how many introgression events, how many admixture events, try to extrapolate where they occurred and when they occurred. So that's something that will be going on in parallel with the projects that we're doing. And it all relies on these genomes that we're generating in Wallacea. So I've probably gone way over time, and I apologize. But I'm really excited to be able to come here and to hopefully collaborate with some of you in the future. There's just so many interesting questions out there that we can only really get at if we combine forces. And that's what I really hope to do. So last slide, that's the people on the paper. A big thank you to the Eichmann Institute in Jakarta, who made all of this possible. And that's led by Professor Herawati Sudoyo. So she runs the project. And yeah, this will be going on for many, many more years. So yeah, that's it. Thank you.
Thank you very much, Ray. That was a brilliant talk, and what is very clearly, as you've shown, a very fast-moving field with some fascinating results coming up. And as you've shown in your conclusion there, as well as generating fantastic data sets and conclusions, it raises more questions for us to test as well. So I'm looking forward to seeing where this goes in the future.
Yes.
I will open up the floor now for questions. Unfortunately, I couldn't bring everyone across to the panelist page. So if you do have a question, please raise your hand and I'll just unmute you and we can have a bit of discussion going forward.
I don't mind hard questions, by the way. Because they're generally the more interesting ones.
I had a quick one for you, Ray.
Yeah.
With your second peak there that you showed across, very prominently in Wallacea and New Guinea, but less so in Australia, the peak at around 3,200 or so.
Yeah.
Up in the north, it might be correlated with Austronesian speaking people, but perhaps less so in Australia. Are there some alternative conclusions or some thoughts about what might be driving those population dispersals?
Yeah, it's interesting. So that 3,000 year peak in Australia, which is quite distinct, was based on just two of these geographically exclusive clades, and they were both contained Torres Strait Islanders. And then when you look at, okay, so who's that? So that seems to be saying something about, in the Torres Strait Islands at that time, there's two samples is not really a radiation, but, there's something going on there at that time. And perhaps that was related to the movement of what looked like effectively, because where they actually meet in the tree, the next group out are Papuan. So what looked like the movement of Papuan mitochondrial lineages into the Torres Strait at that time. So those people draw ancestry effectively from mainland Papuan ancestry. But it's interesting that, that could have happened at really any time, because if you go back, obviously 8,000 or 9,000 years, it was a contiguous landmass. But what we're seeing is 3,000 years ago, when it was an island or a series of islands, that's when this event occurred. And whether it was related to the Austronesian movements or something independent of them, I don't know, but it's just interesting that it comes at exactly the same time. The other interesting thing is you see a similar peak. We don't show it here, but in the Bismarck. So, the islands, Melanesian islands to the east of mainland New Guinea, you have similar patterns. And that's what makes me think the fact that we're seeing it in Wallacea, we're seeing it in the top of Australia, not in continental Australia, but who knows? We've hardly started looking in Australia, and out to the further to the east, all around about the same time. It just makes me think that there's something going on that is probably connected. And whether that's the introduction of these technologies, whether it's because Austronesians were mixing, and we know they did mix right around the lowland areas of New Guinea, that mixing was fairly extensive. Interestingly, did not get up into the highlands at all. So they were pretty much isolated at that point. And doesn't look like it got into, at least at the stage, into mainland Australia, especially away from the coast. So yeah, I have no idea. I don't know much about these sorts of processes. I don't have any sort of intuitions on that, but it seems like that these processes could be related.
Thanks, Ray. We've got a question from Jim.
Yep.
Oh, that's it. Thank you. That was a great talk, Ray. Thanks very much.
Oh, you're welcome.
I want to ask a couple of questions, but first I want to focus on the recent papers coming out on the early populations in Vanuatu. I don't understand genetics at all. I'm an old fogey. I think I grew up before they invented genetics. But in the results out of Teuma Cemetery, I think it is three individuals claim to have if I use the word pure, a genome which is not adulterated, if that's the right word, with Papuan genes that they're East Asian.
Mm-hmm.
So what are you familiar with this material?
Yes, not. So I remember a paper from 2016. I think it was from the David Reich's group over in the US.
Yes, but that was one of the first of the three. Now, what puzzles me is how would people move through Wallacea, well, through island Southeast Asia, and then much of island Melanesia without being intermarried with other populations.
Yeah, because I think, I'm trying to remember now what that paper was showing, because they had a handful of genomes, right? But this is the power of ancient DNA, that's incredible. So they had maybe 5 or 6 genomes from, was it Fiji, Tonga, Samoa? Were there any from Vanuatu in that study?
Yes.
There were. I just remember they had, and I might be wrong here, there seemed to be a movement, initial movement into those areas where there was no obvious Melanesian ancestry. It was effectively the Austronesian seafarers who arrived there. And so these ancient individuals draw ancestry only from those Austronesian seafarers. But then there was a subsequent movement of Papuan-related ancestry. And I think they've actually, in subsequent papers, worked out that this is somewhere in island Melanesia as the most likely, where if those modern populations are a good proxy for the ancient population, then island Melanesia would be the place where those people were coming from. But that was subsequent to the initial settlement.
Yes, that's correct.
Yeah, so I guess, I mean, the most simple explanation would be that those initial movements were of Austronesian seafarers that didn't have any ancestry, at least, from any Melanesian component. So I can only sort of use my imagination to try to think what those movements would have been like how long they had to stay in a particular place before moving to the next place, and how often they would interact and mix. And, you know, there could have been, it looks like from this Philippines paper that came out a few months ago, the Austronesians were themselves a pretty complex assemblage of ancestries. So I think they were probably mixing, but differentially mixing. So they weren't just bringing Austronesian ancestry, they were bringing other ancestries in with them in different amounts and probably at different times. How well we'd be able to reconstruct that is anyone's guess, but with lots and lots of genomes, we'll give it a good shake. But it seems to me like that might be what has happened. I must admit, I haven't looked at the subsequent papers, and there's been a few. And ancient genomes, you can get a new ancient genome, and it can change how we've thought about those processes in the past.
Oh, thanks.
No problems. And by the way, I'm working with Gludug, who is the PhD student on this paper, is talking to with Hera Sadoya, his boss, to archaeologists all around Indonesia, and she really wants to start up an ancient DNA lab in Indonesia and not have to ship it off everywhere around the world. And so it's really opening possibilities for doing ancient DNA research right across Indonesia and also into West Papua, obviously, which would be pretty amazing, because there's just so much more you can do with ancient DNA. Yeah. I don't know what's going on in Vanuatu. That's highly competitive. I mean, that's, you know, you've got the Max Planck in Europe, and obviously we're putting up papers side by side. But yeah, I mean, you'll see a lot more on that space for sure over the next few years.
Okay, thank you, Ben.
Yeah, you're welcome.
Ray, just got a couple of questions here in the text. First one here is, any relationship 3,000 years ago to volcanic activity on the Ring of Fire?
Oh, I don't know. I hadn't even thought of it. Yeah, I guess that's plausible.
And a follow-up question here, could your 3,000-year-old Papuan influx to Torres Strait can accommodate previous ideas for an influx 2,600 years ago?
Yes, the dates will have what I didn't show you there is the error margins where actually if we go back, they are effectively error margins. So you can see if we were to build a distribution based on those overall sort of patterns, you're going probably back to 5,000 and there's up to 1,500 years for each of them. And that would still be pretty reliable. So you can get more accurate dates when you start looking at full genomes, but it comes also with more complexity too. But yeah, this is where the archaeology is really important, and other lines of information. Linguistics would be another one. The fact that at least in these regions, you can probably get a fair idea when languages first moved into a region, or maybe not. Actually, I don't know how easy that would be. But really, genetics will take you so far. But it's putting points on maps, right? That's what we do, and making inferences. It's completely agnostic, and that helps in sometimes of other sources of information. But there's no reason why we can't start developing models which incorporate that information, and even quantitative models that combine them to come up with particular inferences. I mean, that's going to be a real challenge, but I think that's where people are starting to look now.
Interesting. Any further questions? I think that's it. Thanks very much, Ray, and to everyone who's coming along. Again, a huge thank you for presenting today. I think it was very clear, despite the complexity of the background of genetics, and very fascinating as well. So I really appreciate it.
Yeah, no, you're welcome. Thanks everybody for coming along, and hope to meet some of you face to face before too long. Maybe with a mask on.
Yeah, here's hoping.
All right.
Cool. Take care, everyone.